going to turn this right over to Susan Brown, who is the leader of the apple breeding project here and a number of other things. Susan and I, we were just talking, we kind of go way back here because I can remember the first time I met Susan was at a reception that the dean was having for new faculty. And I was plant breeder from Ithaca, and I was there, and I met Susan, who was a plant breeder from Geneva, and she was there. We were both new faculty at the same time. So anyway, we've known each other over the years, and it's a real pleasure to introduce Susan to you, and she'll tell you what's going to happen from there.
Thank you. Welcome. I'm very glad that this group could attend today, and I'd like to, I'm curious, how many have never been to the station before? It's high time. So welcome. It's great to see some familiar faces, friends and neighbors, colleagues, and I hope today I can share some of the passion that I have and all of you have had for your particular crop. I think apple crop, sorry, it's a bias, whatever sphere of research that you do. There's so much with improving a variety or an area. And what's nice about my position is I get to be a jack of all trades and hopefully an expert in some, but I rely on the kindness of my colleagues. So today, Cornell apple breeding, you'll get a chance to taste the apples of the future. I am associate chair of the merged departments of horticulture. What Mark didn't say is he mentioned the 4 departments, but last year in August, all 4 departments merged with their sister counterparts in Ithaca. So Marvin Pritz is the chair of horticulture, which spans both locations, and I'm the associate chair. And we don't divide it up that I'm here, I take care of the group here, he's down there, but we've integrated it, and I think it's enhancing the opportunities for collaboration. I'm also pleased to be the Herman M Cohen Professor of Horticulture, and I lead a tree fruit genomics initiative, which very simply is using all the latest techniques to tweeze out genes of importance, not necessarily now, but for the future. But especially in front of this group, I think it's really important that I recognize my predecessors, Dr. Roger Way and Dr. Bob Lamb. And I don't know the origin of this quote. I heard it's a Celtic saying, but it's one of my favorites. We have all drunk from wells we did not dig and have been warmed by fires we did not build. And I know that I benefited so much from the generosity of people like Jerry Van Buren and Malcolm Bourne when I came to the station. And I'm hoping to follow that same tradition that Roger and Bob graciously let me be the expert, even though I might not have been the expert at the time, and showed me the potential. I'm hoping to do that with students and graduate students and postdocs. And I think we have a wonderful legacy, and it's my honor to maintain that. So the apples of the future, something that happened last year which was phenomenal. When I came and started, I came in 1985 and I switched to apple breeding in 1990. And in 1990 there were 28 genes identified in apple, which we were thrilled with. It was more than a lot of crops, but once the genome was sequenced, we now know that there are 57,000 genes in apple. And that genome sequence really allows us to find characteristics that are important to consumers and growers and use techniques that we never thought would be possible. So all the techniques that are being used in the Human Genome Project, I can use now. And it's in large part to this rose breed grant, which apple is a member of the rose family. And I always go nuts because some researcher said, well, that's because you can smell the rose in an apple. I disagree with that assessment, but pears, apples, peaches, cherries are all members of the rose family, and we can find genes. There's been a gene found in peach that was also found in rose and in apple, and it's located in a very similar part of the genome. So this funding allows us to use modern tools and train the next generation. Okay, ancient apples. Most of you probably know, but the trivia question is, was the apple in the Garden of Eden? How many say yes? No. So they believe now that I think it was a fig or a date because just the origin and the location didn't work. But that hasn't stopped us from claiming that for eons. The center of origin for apple is in the Kazakhstan, Kazakhstan, China area. We've been lucky to have researchers go and get apple from the center of origin. Anyone read The Botany of Desire? Okay, we get requests constantly to go to the Kazakh forests, and it really is a wonderful book if you haven't had a chance. It details apples, potatoes, marijuana, and tulips. And the New York Times reporter said that the chapter on marijuana was, interestingly, the most lucid, which I thought was an interesting comment. Apple is an ancient polyploid, which most plants are. So that means two different genera of different chromosomes came together. We have 25 species, which we don't use enough of. Apple has 17 chromosomes in a haploid cell, and it behaves as diploid, which is wonderful in breeding. It just means that the segregation is much more simple. Okay, so you've heard the story as American as apple pie, but the apples that the settlers found when they came here was Malus fusca. And I think Thoreau or Emerson says that it's sour enough to make a jaybird scream. That's what we had. That was our American apple. But when the colonists brought seeds or whips or saplings, there really was an American melting pot. Johnny Appleseed was a real person. There's some interesting histories. He was a rather strange person. But he did spread apple seeds everywhere. And when those apples fruited, the best and the hardiest survived, and they intermated and produced new hybrids. And that's where our whole industry started. But when you look at heirloom varieties, and there's a big interest in heirlooms, it's because heirlooms have a wonderful story. Thomas Jefferson's favorite apple. And people say, oh, I remember famous from my childhood. And then when you supply famous, they say, it's not quite as good as I remember. Because those apples were bred or developed to be stored. You put them in a storage cellar and you let them survive. Russet types didn't, the moisture didn't go out as hard as it normally would. And the real reason that people planted apples was not for fresh eating, but it was for a safe form of drinking water. And if it was fermented, so much the better. So natural selection. What I do is essentially what a bee does, but I use a knowledge of genetics. And in nature, a bee brings pollen from one apple variety to another. You can't have fruit set without two varieties, so you have to have cross-pollination. But when you cut open, and my mom's a gardener, and Bob, let me know when I get out of the script. When you cut an apple open and you're trying to grow avocados or peaches or apples from seed, we say it doesn't come true. And that's because each seed, if this apple was a northern spy and we cut it open, each apple has 50% of the genes shuffled from northern spy, but the other half is whatever the pollen was. So it could be a crab apple. It could be, so each seed in an apple could actually have a different father. So I actually control so that I know what the female is and what the male is. And then every seed in that apple that I create is a genetic hybrid. And it's just like as you've had families, it just means that I'm hoping that those offspring have the best qualities and none of the qualities that the two parents share. I'm going to switch back and forth a little bit, but I think it's always important to do some statistics because it is interesting and a lot of people don't realize that China is the giant in apple production. And it's going to continue to be the giant. When you look at an apple juice box or container, it's very hard to find US made in that apple juice. They produce a ton of apples, but they don't have the facility to do fresh market production. And so they're the largest. We're very large in the ranks of other areas. So we outdo individual countries, but not total for Europe. And then in the US, we are second to Washington, but you can see it's a relatively distant second. But I will argue that our apples are better, non-biased of course. And we also have more diversity of offerings. Top varieties. We didn't have time to ask favorite apples, but you notice of the top varieties in the US, you don't see McIntosh, which is our #1 variety. Delicious, Golden Delicious, Granny Smith, Gala, and Fuji are the lion's share, but you're seeing more and more new varieties. Delicious and Golden Delicious are not related at all. It was just the advent of marketing. Delicious was a good name, well, Golden Delicious would work too. And so we still see kind of a monopoly, but that is changing. There's tremendous genetic diversity at Apple. I'm like a kid in a candy store. So we have the repository where we have apples, and we needed to get light to show the video. Is this enough light for people? Okay. So we have apples like this that you can make one pie out of. And we have crab apples that might have genes for resistance. We tried to set the world's record for the largest apple, my family and I, but it has to be 4.8 pounds, which is a big apple. But that genetic diversity and the ability to use all of this material funded by the government is wonderful. Okay, so we have tremendous variations, and we have people say, could you tell me what this stuff is? What is this long? You don't usually see an apple that looks like this. Or these are some of the Malus species. These are all new hybrids. This particular apple, even though it looks lousy and is small, I think kids would love. We call it Sour Snap. It has enough acid and enough sugar to rival those sweet candies that your kids and grandkids love to eat. But it's natural, has high vitamin C. So all of these things are new hybrids. They don't have a name. They're new creations. Some of them are not going to make it because this, even though this is intriguing, it gets a greasy skin. But we can change anything. I can make a dark black apple. I can make broad peppermint stripes. But I'm interested in the flavors and the textures. Spicy. We have an apple that wasn't ready that has a hint of anise. If you like anise, you love it. If you don't like anise, obviously you don't. But if I gave it to this group, 50% of you would like it and 50% of you would not. It's really interesting. We have textures. Malcolm Bourne is an expert in texture and helped ignite my interest in that topic and cherries to begin with. But we found out that I could breed apples that are too hard. I had to go to the dentist because my teeth hurt. And I said, I'm biting into apples that are 25 pounds pressure, where the apples you get at the grocery store are probably 14 pounds pressure. It's like biting into a bowling ball. And he said, well, why are you biting into these apples? Because you don't know that they're that hard. So genetic improvement can go sometimes a little too far. And so you need to get it firm enough for consumers to like, but not so firm that you don't want people to have difficulty eating it. We can do resistances. We can change the sugar content. And a person will like an apple if it has 13% sugar or degrees Brix. If you raise it up to 14, they like it more. But if the apple isn't sufficiently firm, you can raise it to 16 and it won't matter. So we study what consumers want and see what we can change. We're well known. We have 66 apple varieties, and we're well known for Cortland, McCowan, Empire, and Jonagold. If you haven't tried a Jonagold, it's considered one of the world's best apples. But it took off in Japan prior to being popular in the States. Okay, so a little bit of facts about the apple breeding program. We're one of the oldest in the country. We're one of the largest in the world. I'm second or third largest behind New Zealand. And if I could have their budget, boy, could I do more than they have. And it's a wonderful program. I have to be careful because I'm not usually taped and I tend to be fairly candid. We're one of only 3 apple breeding programs in the US, and one of them was only formed in 1994. When you go to Japan, they may have 12 apple breeders in a country of that size, so it really is a different magnitude. And all breeding programs throughout the world are starting to erode because of a lack of funds to support them. We are using primarily traditional breeding. And I mention this because the Wall Street Journal talked about apricot plum hybrids and talked about our program. And people commented that we shouldn't have frankenfoods and we shouldn't play with nature. But these are traditional, just like early plant breeders or farmers selected the biggest ear of corn or the biggest pumpkin. This is a natural process. But we do genetically modified. And we believe in that technology as well. So how do we make new varieties? Every spring, we're in a panic. Apples are starting to blossom. They're at this stage called the popcorn stage, which is popcorn bloom is when I need to get it, before the bees get to it and bring pollen. So I have to go out with as many people as I can get, and we use our fingernails to pop off the petals. We do that to keep bees from visiting. So now we have what is called an emasculated flower, and we collect pollen from another parent, and we apply that pollen to this seed parent. That ensures that every seed from that cross is a hybrid that shares those 2 parents. And we'll plant thousands of them. And what we want is the good characteristics without the bad. So again, I'm going to emphasize this is genetics versus GMOs. This is genetic manipulation, and the gene gun was invented next door, which uses the mechanism of a gun to hurl DNA into a cell and can transform it. That is genetically modified or biotechnology, and we do that here because you can do some trait improvement with that you can't do by traditional breeding. Family resemblance genes. I always tell groups that if you're a parent, you've done genetic modification. And sometimes they do get those genes from your spouse that you will tease about. This is my offspring. I have thousands of apple seedlings, but I'm most proud of these 2 offspring. She's a sophomore at Cornell BEE, and my son is currently a junior at Finger Lakes Community College. But you can see there's family resemblance between them and me, between my husband, and it's the segregation that we're looking for. And unfortunately, stubbornness is inherited, and they got double doses. Okay, so we choose 2 parents. We can choose 2 red apples, and if they have a gene for yellow, we'll get yellow fruit. So this is 1 apple from different seedling trees of 2 different crosses. In the first cross, you can see we get segregation for color, but we get a lot of segregation for size. Some big apples, some little apples. This cross size is very uniform, but what's amazing about this cross that you can't see is it segregates for texture and flavor and vitamin C content and non-browning. So this is the segregation that we can look for. So every year we plant upwards to 10,000 seeds. And we plant them in the seedlings fruit in about 4 years, which is pretty amazing. One of the apples that I've named has been 11 years from my making the cross to it being commercialized. It used to be 40 years. And then we have to find the promising offspring. So I broke an important rule for this group that I don't usually break, and that's don't schedule any speaking engagements during harvest time. Because as soon as you guys leave, I will be putting on jeans and boots and heading out to the orchard. We go through seedlings and we look for those that combine the best characteristics. But the problem is we want offspring that don't look like their parents, because the parents have a set marketing. We want something that's different. So it looks different than the parent and it has some different characteristics. This is going to be a promising selection. This one isn't because it's ununiform, it has disease. And when we think we have something, then we propagate it. So I've explained to you that I make seeds, but apple is a clonally propagated crop. If you go to an orchard of McIntosh, every tree of that McIntosh dates back to the early 1700s to a tree that was discovered. So when we find a tree that we like, we take a bud from that tree and we put it on a rootstock, something that's been bred with somebody as excited about it as I am about the fruit. He's as excited about the root system. And we put a bud in, we seal it, and then when it's healed, we cut this off and that becomes the tree. Whoops, sorry. And so every tree in an orchard has a rootstock, a scion, and a union. So if you see apples in the orchard in your home, you'll see a swelling. And that's where those two parts are made. And then I could make hundreds of trees of that one advanced selection. So that's what I did for New York 1, which is right here. And I told, there's going to be 900 acres of these two varieties planted. Some growers signed on without ever trying New York 1 and 2. So I snuck some aside from you because they're hard to come by, and I hope you'll agree that they're good. So New York 1 is got tremendous pressure, 17.9, hard enough, but it's not going to break your teeth. 14.6 bricks, which is really good for an apple variety. And this probably convinced more growers to sign on to this than anything else. That is a productive apple, producing trees at a high density like we do now. Okay, selection 2 is a hybrid of Braeburn by something called 674. And Cy Lee discovered that 674 has resistance to flesh browning. And I made this cross to get resistance to flesh browning and high vitamin C. But it's a really good apple. And if you like a little bit, this is a sweet, mild apple. That is sweet with more of an acid balance. And it did very well in consumer tests. So we had these two great varieties. And in the past, when Roger and Bob were here, at first we used to just give them to the industry. And then we started with the Patenting Act charging a per tree royalty. Well, nowadays, everyone is owning varieties. And they sign exclusives. But the difficulty is some of those exclusives have been with an industry's competitor. There's an example where a blueberry from one state was sold to a competitor in another state. And as you might understand, the administration at that university got a lot of phone calls. So we wanted to find a partner to do an exclusive because we felt they were good enough. But the problem we faced was we have 674 apple growers. That's a lot of people to get together. We're second in the nation. McIntosh is our #1 variety. But what helped us is that many of our growers couldn't join existing apple clubs. And let me explain that an apple club is where you control the supply, because if you have oversupply, the prices go down. And you also provide money for marketing, which is an important part, and you get enough volume to satisfy buyers. So our growers were not able to join in on some existing clubs. And our apple production is scattered throughout the region. We're lucky to be in the heart of really good apple production, but we also have it in Long Island, in the Hudson Valley, in the Champlain. So what happened is Sweet Tango, Honeycrisp. Any Honeycrisp fans here? Okay, so New York 1, if you like Honeycrisp, you have to try that. Honeycrisp is a wonderful variety, but only 40% of the fruit that's produced is good enough to sell. That's a tremendous economic loss to growers. So we had New York 1 that was like Honeycrisp, but doesn't have the disorders. Well, Minnesota had something like Honeycrisp called Sweet Tango. And they thought it was good, and they offered it to growers in their state. And they only have 200 growers in their state. But they gave rights to one single grower. And so he set up a very nice cooperative of 45 growers throughout the state. But there were some smaller guys in Minnesota that said, this is a state program funded by state dollars. How can we not have access? This isn't fair. And they filed a lawsuit at a local and federal level. Now, I'm friends with the breeder there, I'm friends with the company, but I loved the headlines, and I'm glad that it wasn't my apple that we were talking about. So Sweet Tango headlines: takes more than 2 to Sweet Tango. Use hot new apple already made into a tort. That's my favorite. And apple growers are sour on the university. So when we were looking at how to form this marketing company, this was weighing heavy on our minds. So we had a discussion about, and I never thought as a geneticist that I would be looking at marketing plans, but how do we capture value for a new variety without alienating the industry and getting buy-in from 674 growers? And so we formed a task force. We studied the past. I spoke. We needed an entity that didn't exist. I could have given it to one large grower, but that wouldn't be fair. We'd have haves and have-nots. We needed grower buy-in, and we needed a strong marketing program as part of it. So we gave a call to the industry. And as you can tell, I tend to be fairly candid and fairly blunt. So it was, if you want this apple, you need to step up to the plate. We did editorials. We did fruit school presentations, articles, emails, and meetings. And after it was over, they said if somebody said they didn't know about this, they had to live under a rock, because we put out the call. And I'm pleased to announce that you probably saw something in the Chronicle that we now have a partnership on these 2 varieties with the New York Apple Growers, NYAG. We're equal partners, sharing equally in risk and rewards. Every grower in New York State had the opportunity to join this cooperative. So it's a managed variety, but it's not exclusive. And the industry was thrilled, and we were thrilled. It took a lot of time, but Commissioner Pat Hooker at the time was behind it, as was Cal's administration. And I snuck these apples for you, but you can bet the Board of Trustees are getting some next week. So we have new apples for the New York industry and for consumers. And we really do feel that we're going to offer them superior flavor, crispness, juiciness, firmness, storage and shelf life, freedom from storage disorders without needing to use storage treatments. And that's what it's about. We've done a lot of consumer testing. And then the important thing that you know, because right now homeowners cannot get these trees, and that's not sitting well. In the future, we will allow that. But we're going to have open releases too. Where a grower in Massachusetts, if they want to try a New York apple, we'll have apple varieties that will be available, because it's not fair to shut out the US industry. And we will be pursuing international rights to some of these. So, if you could change anything about future apples, what would it be? And I always love asking kids, because kids say, make it taste like a banana. Make it soft because I don't have front teeth. College kids ask for no core. And I said, come on, no core? How lazy is that? And my son, who's 21, said, that would be cool, Mom. No browning. Any not on that list from this group? Anything that bothers you? Women tend to say, too tough of a skin.
No worms.
No worms. and we can't breed for resistance because we tried to. And apple maggots will try to lay eggs in plexiglass. So if I produced a resistant apple, I'd be going to the dentist and you'd be going to the dentist. Okay, so I want you to understand, and this is the fun part of the talk now that I've gotten some of the background set. It's more than just sampling apples. So somebody once said, send me your best apples, and they hadn't fruited yet. And I said, well, they haven't fruited yet. And he said, well, just let me know which ones you think are going to be good. And I said, if I could do that, I'd be buying a lottery ticket. You can't do a Ouija board. You can use genetic markers, but a lot of it is you have to sample. But we have the ability to look at titratable acidity and tell you how much acid is in that variety. We have a much more sophisticated method than this penetrometer in food science, but we're also getting one ourselves that shows how firm an apple is, not only on the outside, but on the inside. We have DNA markers that I'll talk about that tells you what traits are in an apple before it even fruits. And we have genetic maps that tell you what genes are located together. So if you want to get a redhead in humans, if you wanted to get a redhead without freckles, how many offspring would you have to get that combination? So DNA markers and selection, what we do is we can grind up young leaves, subject it to different DNA tests, and if the presence of a band in a seedling correlates with the presence of a trait, we say those traits are linked, and we can use that molecular marker to screen those seedlings for, let's say, disease resistance. And this is where it would be very useful. This is storage scald. You never see this, but the growers put apples in storage that look wonderful. They take them out and they look like this, so they have to throw those out. If we had a marker that could eliminate those seedlings that tended to have scald, it would be very useful. So I stole this from Ralph Scorza, and I'm crediting him because I thought it was a great analogy of molecular markers. As a breeder, I can look at red and yellow, and in this case, we're looking at red leaf and green leaf. And the cards are marked because we know that they're red or green. And we shuffle the deck, but we can see the segregation. What happens when you have a trait that you can't see, like sugar? So here we have parents that have different traits, but we don't know what they are. And when we shuffle them, we still don't know what they are. But what if a molecular marker allows you to mark that card? And so let's say this is fruit firmness. Then we can shuffle that deck and we can make offspring and we can say, okay, I want this seedling that has disease resistance and flesh firmness. And we can use those markers to select only the best seedlings. And it's powerful technology. We use this for resistance to apple scab. If any of you have apples in your backyard, you probably have apple scab, especially this year. What we can do is we have genes for resistance that started out like this. It took about 4 generations to go from this to a scab-resistant, good quality apple. But we can spray with scab or we can use molecular markers. If the apple leaf looks like this, the seedling fruit of that will have scab. If the reaction looks like that, the seedling will have no scab on the fruit, and we don't have to use fungicides. Post-harvest. I wanted to use post-harvest because that's something that we're really renowned for at Cornell. So Dave Blanpede was a wonderful mentor in this area. I brought him up and I said, I want to know how to look for storage disorders. And he gave me a protocol and he cut open a lot of apples and they were disgusting. All sorts of disorders. And this is where you see a glass half full versus half empty. Because as a breeder, I'm like, I have diseases. And Dave Blampede's like a kid, and he said, Susan, I've never seen so many disorders. And he was thrilled, and I'm glad to make you happy, Dave, but how do we not get these disorders? So we've come a long way. Bob Smock really innovated controlled atmosphere storage, and Bob, if any of you knew him, was a wonderful person. He slipped me a note while I interviewed at Cornell, saying he hoped I got the job, but his vote didn't count at that point. And he was just a wonderful man. And then Chris Watkins is following in those fine footsteps where he's looking at storage disorders with some of our stuff. This shows you if an apple's cut and it's put into a mixture of iodine solution, if it turns black, that means the starch isn't converted to sugar and it's not ready to eat. When it's fully converted, that's a tree-ripe fruit. Growers are going to pick them at different stages of maturity because they get longer storage. And this is a simple test. It's not ideal, but it does give us a measure of how firm or how ripe an apple is. Okay, so some of the fun things that we're doing that I'm proud about. We know that browning is an important trait. 674 is from our program and Liberty is from our program, but 674 was discovered to have a gene for non-browning when equipment broke. It was being processed, it stayed out cut, didn't have browning. So we've done a study and we know that that trait can be transmitted to future progeny. and this is important, not only for juice production, but Happy Dippers, little apple slices of McDonald's Happy Meals. When McDonald's started buying apple slices for their Happy Meals in response to the childhood obesity, they became the largest purchaser of apples in the country, buying 55 million pounds. Well, recently they've decided that each Happy Meal, whether you want to substitute the French fries or not, is going to have apple slices in it. It's going to revolutionize the market. So if we could have apples that are suited to that market that don't need to be treated with anti-browning agents. So it's value added and convenience. It's school lunches. USDA says half an apple is a serving for daycares. So slices you can get in where you can't get a full apple. And we want these traits to be stable and safe, which they are. Okay, so then also, we have wonderful people like Siley and Ruhai Lee that can do antioxidants. And every apple has a ton of antioxidants, but it's determining which one is best, what method is best. And to be honest, I tend to sometimes be a simplest person. People understand vitamin C. Maybe it's not the most important antioxidant, but we all understand vitamin C. So I asked Siley, how much vitamin C is there in an apple? And he said 5 to 10 milligrams. And I said, how much variation is there? And he said, well, we'll look. And some of ours were higher. So I crossed a high vitamin C by a high vitamin C apple. And you can see that McIntosh and Gala are below 5 milligrams. And this is just a random sample of several of our selections. And you can see that we're getting much higher vitamin C. So we'll make the next round. I think we'll easily have as much vitamin C as an orange. And it's natural, and people understand it. Evaluating crispness. I should defer to the expert here, but we've done some interesting work with crispness. Because how many of you knew that if you bite an apple, if you take a section from an apple and bite it in one direction, it's different than if you bite it in another direction. It's called anisotropy. And these are wonderful graduate student projects because they have patience. So one student took cubes of tissue in the right orientation and measured the force needed to propagate a crack. And that correlates with crispness. Other places are doing acoustical, because the sound of an apple is correlated with crispness, but people's mouths different, how they bite apples is different, their teeth structure. So this was one way where we were able to quantify crispness. Tree architecture. We all know what trees in the backyard are supposed to look like. This is what high density plantings look like, where there are easily 1,000 to 3,000 trees in a small area. But what about a tree like that? It's a natural mutation. It's an apple tree without branches, great for small yards, planting against your driveway, garage, and resistant to diseases. So we have these. And I knew it was a hit when my mom said, oh, I want one. But to be a geneticist, I get to, Bob Lamb was quoted as this, but I get to create things and taste new combinations that no one else has ever tasted before. And that's wonderful. But I also get to play creator a little. So what happens when you have a dominant gene for columnar and a dominant gene for weeping? Any idea what's going to happen? Bill? Neither did I. And we see some hybrids that are columnar on top and weeping on the bottom. Both genes are expressed. They don't know what they want to do. And we see segregation, and this is just purely good for the spirit because they're ornamentals and they're beautiful, and we're learning at the same time. This is not a prune tree. It is 1.5 inches off the ground, and it has spokes like a wheel. And we think that that is a mutation for a plant hormone. And so we're starting to do more with other researchers on plant hormones. What about an apple tree like that? Its sister is 5-6 feet high. We could have bonsai apple trees without the work. These are two sister seedlings again. Same age, same genes. One has a gene that we think for a different combination. So we're getting self-fruiting trees without the need for a rootstock, which would cut cost. This is a columnar tree, maybe 1 year old, already fruiting. And we learned that we could grow disease-resistant columnar trees in cages, and we could have the top of the cage open, and insects didn't know enough to hit the cage and go up and over. So we could grow completely pesticide-free trees. And anyone that lives in upstate New York, it helps against deer too. Fruit color. We are using genomic tools to understand fruit color like never before. This isn't a photoshopped apple. It is a chimera that has half red, half yellow. But that helped us uncover what is causing genes for yellow or red to be turned on or turned off. And I do this because I think we're missing the mark and we need to get some Cornell big red stickers for our apples. If you put stickers over apples, the sun doesn't get on, you get this nice logo. In Japan, they do it with good health and they charge $4 an apple and people are happy to do it. So, and then we could also create red-fleshed, high antioxidant types, but right now they're edible, but they're not palatable. Okay, biotechnology. I'm going to just do a little bit on this because what biotechnology is good for is if you like empire. I can do things with breeding, with vitamin C, but I can't give you a higher vitamin C empire. I can give you a new hybrid. So biotechnology allows you to change an existing variety, which has established methods. And we're lucky in apple because just like African violets, we have good regeneration systems. And so we can take plants, put a gene from apple or for another organism in, and this is a case where these are genes for resistance to fire blight, which is a very bad disease for us. It's a bacterial disease. So we do this, we have tests. Herb Baldwinkle is doing this at Geneva. We have field testing under all the proper rules and regulations. And I think that we should not dismiss the technology, but I think people should have a choice, and so that it should be labeled. The gene gun, as I said, was invented here. Dennis Gonzalez was a professor at Cornell. He was smart, he was from Hawaii, and he decided, I'll work on papaya. Not exactly a New York crop, but one that would allow him to go back to Hawaii. But what was amazing is he found the ability to put in genes for resistance to a virus, and he saved the papaya industry. And he gave that technology to a lot of people that were having a papaya tree or two in different countries that was their livelihood. So this is an example of biotechnology that was for the greater good. So my daughter, the sophomore, is upset. She's class of 2014, and I've used this slide, but he was born first. This is, every breeder has quality control at home. So food safety, what we're doing, and so we need to have apples that this was a candid shot of enjoying an apple, and if we could get juiciness and delight like that. So we have apples that are a real treat to eat. They're juicy and they're crisp. We have interesting flavors. And we use a wine wheel to detect flavors. And I tease my colleague that works on cabbage because cabbage is between wet dog and burnt skunk, or a burnt match and skunk. I have a much broader palate, so we have cherry, raspberry, green pepper, but we have a lot of off flavors. And amazingly, we do have to sample them all. If they look good, we have to bite and sample. And somebody asked me what apples I use for pies. I don't bake pies. A lot of apples. We have a very vibrant New York industry, and by this partnership, that industry is going to continue and flourish. So I'm very happy about that. We need help for names. New York 1 and 2, they're doing some marketing classes at Cornell are working on it. They are hiring experts in names because Honeycrisp was a great name. There's other names that have been awful. A new delicious sport was just called Adam's apple. And I'm going to have to change this when we post it on the web, but I used this before. Arnold Schwarzenegger, in Austria, they have the Arnold apple, which I thought was a bad idea, but it's even more of a bad idea given recent events. I can't possibly acknowledge all the people that have helped me in my many walks of life, and so this is trying to do a bit of a job on that. We do receive funding, but apple breeding is very expensive, so we are looking at innovative ways to get the job done. And I hope I was able to share, one, my enthusiasm for apple, what the potential is, and the next time you try an apple or one of these, that you think about it in a little bit different way. And I thank you very much for the opportunity to tell you about it today.
Well, thank you very much, Susan. We have part of our Cape tradition is to give a gift to the
thank
speaker,
you.
so we thank you very much.
Thank you very much.
I should have said at the beginning that the reason we came to Geneva this week or this year was because Malcolm Bourne and Hugh Price and a couple others accosted me down in Ithaca at one of our Cape seminars down there, and they said, I should move over?
No,
Oh,
stand up.
yeah, okay, yeah, will you stand up and see? Hugh isn't here, I don't think.
No, Hugh's traveling. Malcolm?
Yeah.
I was the one who suggested you all come up here. That was it. Susan and Tom Burr and the rest did all the work. So, but I thought you people ought to come up and see what we do at Geneva. A lot of exciting stuff going up here. So I'd like to say welcome too.